Right. So after we had gone over Punnett squares and different types of problems and test crosses, we did do a couple of little labs. The first one was lab 20. You should make sure that you get a copy of each lab. If you were absent for both days, then I'm not really too concerned about making sure that you hand them in. I'll just say that you were absent all week because you were sick, but you are still responsible for understanding what was covered. So I'd still suggest that you kind of look over it with someone else, make sure you understand what was done, and especially focus on um, the pedigree analysis parts, okay? Because you're going to have to be able to analyze pedigree charts. So the first one was lab 20. It was the probability lab. And basically we used pennies in order to represent the gametes. Okay, we put, um, so if we had two pennies, then we had one penny represent the, the possible gametes from the mother, and the other penny represent the possible gametes from the father. Since each parent was heterozygous for albinism, so they had a big allele meaning they were normal, and a little allele that was the recessive trait. They had a big S on one side and a little s on the other. Big S for normal skin, and little s, if they ended up with both little s's, then they'd have um, albinism. Okay, so this is based on probability. Hi, thank you. I just got my menu order. Um, People coming in to check on me all the time. Okay, so this one was based on probability, and this was just a slide to review how we look at probability. So what's the probability of flipping a coin and getting a heads? We could look at it as in terms of fractions. We look at the number of desired outcomes over the total possible outcomes. Okay, so if you wanted to flip a coin and you get heads, then your chances are 1 out of 2. So 1 is your desired heads out of 2 possibilities, heads or tails. You could write that as 50 to 50 or 1 to 1 or even 50%, express it as a percentage. The product rule we've already used in a couple of problems. Basically it says the probability that two or more independent events will occur together is the product of their individual probabilities. So that was like when we looked at the probability of having a normal child and then one with albinism, and then for I think it was number 13 when we went over that problem. So we said it was like 3/4 times 1/4 times 1/4 to give us the possibility of having a normal and then two albino children. Okay, so this bottom part is just showing the two different ways that you could show what is the probability of flipping two coins and getting heads on each. On the left, it's showing it using the product rule. So you'd have um, the probability of getting heads for one coin is 1 and 2 times the probability of it in the other is 1 and 2. So half times half gives you a quarter. If instead you looked at all the possible outcomes, there's 4 possible outcomes when you flip two coins. You get heads, 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 tails, tails, heads, or tails, tails. So if you looked at how many of them are heads, heads, then you'd get 1 in the possible 4. Okay, so this is just a summary of probability and how we look at them. So in the lab, basically they flipped the coins 100 times. Each group flipped 100 times, and there were 14 groups. Okay, when they looked at their own results, then some of them had very close to what was expected. Okay, so in that particular lab, we had, let me see, do I have a pen? No, pen. Okay, so we had SS by SS. Those are the parents. Okay, so this was the Punnett square on the first page that they had to fill out. They had big S, big S, big S, little s, big S, little s, and little s, little s. Because on each penny, they had a big S and a little s. The mom chance is big S and a little s, and the dad, they could get a big S and a little s. So they looked at, okay, if you looked at the Punnett square, then in 4 offspring, 1 would be expected to be big S, 2 would be expected to be big S, little s, and 1 would be expected to be little s, little s. That's the genotypic expectation. Okay, if we were to look at that in 100 tosses, like what they did, then you'd expect it to be, instead of 1 to 2 to 1, you'd expect it to be 25 to 50 if we looked at 100 instead to 25. Okay, so that's where they got the numbers in their first table. That's expected. So that's what we expect or mathematically figure out using the Punnett square. When they actually did the tosses, then they did their little tallies, like 1, 2, 3, 4, 5. They tossed them 100 times, and they looked at their actual results. So the last column showed their actual results. And some ended up with close to what's expected, 25, 50, 25. But some were kind of off. And that's expected to be, because remember, it's what actually happens in nature is just by chance. So by looking at a single set of results, so for one group, sometimes they weren't exact. Okay, but when we looked at the whole class overall, we found that the totals, when we looked at all 14 groups, um, in the end, when we looked at all 14 groups, then the totals approximated were really close to the 25, 50, 25. Okay, that was for genotype. So if you looked at phenotype, then to be normal skinned, you could be big S, little s, or big S, or big S, or big S, little s. Okay, so of these, it would be 3 normal, 3 chances of being normal, okay, to 1 chance of being, right, little s, little s. So the phenotypic ratio in the second table that they had was 3 to 1. If we looked at in 100 possible offspring instead of 4, it would be 75 to 25. Okay, so that's expected. And when they did their totals, instead of having to toss them all again, all they did was they took their totals for, in the first table, they had the number that were big S, and then the number that were big S, or little s, big S. And they added those together. So if they had, let's say, 21 and 57, then they'd have a total of 78. Okay, so they had 78 that were like that, and then they'd have, of their other tosses, 20, I cannot add 78. So this would be 22. Yeah, 22. So let's say their tosses were 21, they got big S, big S. They had 57 that were big S, little s, and they had 22 that were little s, little s. So in terms of how many were normal or appeared normal, it would be 78. And those that appeared albino would be 22 for their totals. Okay, this would make more sense if I actually had the lab sheet to show you. So you might want to actually get someone to show you their lab sheet. But basically, the whole gist of Lab 20 was to get you to realize that the more times that the experiment is repeated, the closer the observed values are going to be to what was expected or shown mathematically. Okay, expected values are ones that are determined mathematically using the product rule or the Punnett square. Okay, there's handouts that were given out as well on those days. So if you want to check and ask whoever's in for me, whether it's Miss Johnson or I'm not sure what's going to happen later in the week, but you should get those handouts because that will help with understanding those labs as well as the pedigree analysis. Okay, so that was the main gist of the penny flipping part of that experiment. You should do that last question on Lab 20, number 14, where it talks about the different families. They looked at, I think it was 8 different families. In other words, they looked at the offspring from heterozygous parents that were heterozygous for albinism, so they were carriers, and they asked certain questions about them. So remember, when you're looking at a pedigree, the squares are males, circles are females, and if it's filled in, it means that they show the trait. Okay, so try number 14, get someone else to go over their answers with you so that you'll understand what was explained and understand how to do the pedigree charts. Okay, so this was just summarizing the results that they got, and we showed that basically what's expected for this is 1,050 and 350. So when they looked at the class totals and the actual results that we saw when we looked at all of the student results was really close to what was expected mathematically, as opposed to some of them which were like 81-19. They're fairly far from 75 to 25. Okay. All right. So Lab 35, they looked at different mono monohybrid traits. In other words, they looked at single traits that were either dominant or recessive, and they were kind of looking at their own traits and figuring out what their genotype would be. So you can do that for fun with someone. Get them to help you understand what was done. Okay, there were 11 traits that they looked at, and you're either going to be dominant or recessive. If you're dominant, unless you look at your parents, then you're probably just going to put the big letter with a line. Okay, so um, what's most important from that lab is for you to look at the pedigree charts and try and do those, and then go back to someone else and ask them to help explain, or you can ask the substitute. I think I left the answer keys there. If not, then you can ask someone else from class because we went over the answers together. Okay, so this was one of the handouts that was given, so make sure that you get it. Okay, because it's got good hints for how you do pedigree analysis. Okay, basically this one is the question is figure out the genotypes of all the individuals in the following pedigree chart if it shows the inheritance patterns for a recessive trait albinism. The normal skin allele is big S, so the recessive allele will be little s. And it's an autosomal recessive disorder. Okay, so if they show it, which means they're filled in, then they would be autosomal, or sorry, they would be homozygous recessive, which means they'd have to have little s. Okay, so when you see them, when you see them written like this, that means that they are married. Okay, the square is the male and the circle is the female. When you see them attached in this fashion, then these ones down here are their children. Okay, so we have a line coming off, and then they come off of a horizontal line. Um, each one of the ones that's coming off are children. Okay, and they come off in a certain order. So these particular parents had a daughter, and then they had a son, and another son, and another daughter. These two are from different families, but they are joined horizontally, so they got married. Okay, so probably the easiest way to go through this, oh yes, and this is generations. So this would be generation 1, and this would be the second generation, their offspring. Okay, the easiest way to approach them is to go through and fill out I'm just going to get rid of some of that red stuff so it's not in the way. To go through and figure out, okay, which ones do I know the genotypes for? It's asking you to figure out all of the genotypes, but if you go through and figure out, okay, well, I know that the ones that are recessive are going to be little s, little s. So I'm going to go fill them out first. Then I'm going to figure out the rest by deducing. Okay, so I know that because this child has to have two little s's in order to have the disease, each of this girl's parents must have had a little s. Okay, but since they're not filled in, they must also have a big S, okay, because they're showing the dominant trait. So they would have to be big S, little s, in order to give the daughter two little s's. Okay, her siblings don't show the traits, so they must have a big S. But we can't tell for sure because we don't know anything about their children. We can't tell for sure whether they have a big S or a little s that goes with that big S. So to show we're not sure, we put a line. Okay, they might be big S or they might be little s. Okay, but I know they have one big S for sure. Now, over on this side, we know that each of these four children are showing the dominant trait, so they have to have a big S. Okay, but we also know that their mother can only give them a little s, so they must be big S, little s. Okay, and while you might think that because all of the offspring are showing the dominant trait, that maybe the father is big S, we cannot tell for sure. So since we can't tell for sure, we have to put big S with a line to show that. Okay, so that's the general idea for how you do the pedigree charts. You should do the ones that are in Lab 35 and go over the answers with someone who already has theirs corrected. Everyone else has theirs corrected if they were there, so it would be a really good idea to do that because there will be some on your test. Okay, that's the answer for the particular one. Okay, we did on Friday go over the next set of uh, experiments that Mendel had done. Okay, so he did dihybrid crosses next. So remember, mono meant there was one trait being studied. In this case, he looked at two traits and crosses again between two purebred parents. Okay, so in this case, he wanted to see how multiple traits are inherited to see if one trait affected another. So to set up his experiments, he selectively bred, but instead of just for one trait, like tall, like height of the plant, tall or short, this time he did, say, in this case, he did shape and color of the seeds. Okay, so he bred one set so that they were both dominant traits, round and yellow, and he bred one set of plants so that they were both the recessive trait, wrinkled and green. Then he performed this dihybrid cross. So basically he bred the two types of purebred plants, and they were the parent generation. So these guys are the parents. He looked at their babies, their offspring, the plants that were produced, so the F1 or first filial generation, and then he self-pollinated those ones or got them to cross with themselves and looked at the offspring that came about. Okay, so for a dihybrid cross, we have to look at the two letters together. So if it was round and yellow seeds, then they're both the dominant traits, so they're both pure or both homozygous for the big letters. Okay, and he mated that one with one that's homozygous recessive for both. Okay, little r, little r, little y, little I, wrinkled and green. Okay, so again, you're going to take the possible genotype of the parents. You put them along the top and the side, and then you look at the possible gametes. Okay, so in this case, it's fairly straightforward. This parent can only give big R's and big Y's. Okay, so it can give this big R and this big Y here, this big R and this big Y here, this big R and this big Y here, and this big R and this big Y there. So there's 4 ways, but it's always big R, big Y. In this case, there's 4 ways of doing it, but it's always going to be little r, little y. So when you're doing this type, sometimes you're going to see that there's shortcuts, that you don't have to fill in the whole chart to realize what the results are. Until you're used to it, you may want to still fill in the whole chart, but there are some shortcuts that you can use. Anyhow, if you were to fill in the whole chart, you would find that every single time it's going to be big R, big Y, little y. Okay, so it's heterozygous for both traits. So since you have the dominant allele for each trait, they are going to show both dominant traits. All the offspring have the same genotype, so 100% are heterozygous dominant for both. And all of them show both dominant traits. They're all round and yellow. And this is what he found when he repeated it over and over and over. Then, this is pretty messy looking, but then he took those offspring, so the F1 generation, so are the ones that are heterozygous for both, and he mated them with themselves. So that means he took this offspring and he mated it with itself, let it self-pollinate. In other words, he took the pollen and he put it together with the ovules from the same plant. Okay, so here's their genotypes, okay, for the parent. So you look and you figure out, okay, what are the possible gamete combinations? You can have big R and big Y there. You can have a big R and a little y there. Little r and a big Y there. And little r and little y there. Since both parents have the same genotype, then they have the same possible gametes. So I just wrote them the same along the top. Okay, and then you fill in your square. This time they're all, well, they're not all different, but there's many different genotypes. Okay, so when you fill it all in, it's easiest to write your phenotype in the boxes too, and then count them up. Okay, and what he found every time was that he got a ratio of 9 that show both dominant traits, so 9 round yellow, to 3 that show round and green, to 3 that show wrinkled and yellow, to 1 that shows both recessive traits. So that's the phenotypic ratio, 9 to 3 to 3 to 1. Okay, 9 to 3 to 3 to 1. That's just showing the picture of the same thing. Okay, so from his results, he proposed the law of independent assortment, which says that the inheritance of alleles for one trait does not affect the inheritance of alleles for another. Different pairs of alleles are passed to the offspring independently of one another. Okay, so then we did practice one, and we looked at, okay, well, let's see if we were to do a dihybrid cross. Oops, this was the practice one. So again, anytime you're doing a Punnett square, you need to figure out what are the possible genotypes of the parents. Okay, what are the genotypes, and then figure out what gametes could go into making their offspring. So determine the results from a cross between a plant that's homozygous yellow for seed color, so big Y, because it's dominant, heterozygous round, so big R, little r, for seed shape, and a plant that has wrinkled green. So wrinkled would be little r, and green would be little y, little y. Okay, so there's wrinkled and green, little r, little r, little y, little y. And the other one was big R, little r, big Y, big Y. Okay, so those are the parents that are being crossed. Then you break it down to show the different possible gamete combinations. So we have big R with a big Y there. We have big R with a big Y there. Okay, same. Little r with a big Y here, and little r with a big Y here. So again, because because we have a lot of things that are showing the same, sometimes you're going to see that there are shortcuts you can use. If you're not sure and you're worried, fill in the whole thing, okay? So with this parent, there's only little r's and little y's. So there's 4 different ways you can get it, but it's always going to be little r, little y in the gametes. When you put them together, you're going to end up with half of them. So 8 of the 16, if you filled out the whole thing, would be big R, little r, big R, oh, sorry, big R, little r, big Y, little y. So they'd be heterozygous for both, round and yellow. The other half, or the other 8, would be wrinkled and yellow. Little r, big Y, little y. Okay? So you'd express your ratios, same as you did with the monohybrids. I think that should bring you up to speed to what lessons were shown in the videos this week. And if you happen to miss any of those, then just ask the substitute if she wouldn't mind letting you watch those ones as well. Okay, good luck. I hope that it's helped to be able to watch these and get caught up. And if I can help, try and see about sending me a text message. I've been trying to get my husband to check on my um, trying to get him to check my emails to see if there's anything else, but it's a little tricky being here without internet. So I'll help as much as I can, but remember that there are some other teachers there too that you can always try and ask questions of. And ask your classmates because they've probably already asked some of the questions that you have. Okay, good luck and take care. Mrs. Spence, we'll see you soon.